welcome you all to Biotech Made Easy videos. Today I'm going to talk about the Maxam and Gilbert method of sequencing. Let us see what is sequencing. Sequencing is to find out the exact order of bases within a DNA molecule. That means if you have a DNA molecule and you do not know about the order of the bases, then you can find out with the help of methods of sequencing. Alan Maxam and Walter Gilbert in 1977 gave this technique, which is commonly known as Maxam and Gilbert method of sequencing. This method is also known as chemical degradation method, and it is because this method is based on chemical modification and then subsequent cleavage of DNA. That's why it is known as chemical degradation. And in this method, the certain chemicals or the combination of the chemicals is used, and that actually determines the bases. Along with the Sanger sequencing, it was the first method which became popular. Sanger method is another method which also was discovered in 1977, but the principle of Sanger method is entirely different. I have made a video on Sanger method also, so if you want to know about the Sanger method in detail, you can go through that video. So initially these two methods became very popular. These are the sequencing methods, but of course these days many advanced methods have already been introduced, and they are very economic and they take less time. So these days these Maxam and Gilbert and Sanger methods are not that much used. Now we'll talk about the procedure. First of all, you must have a DNA sample for which you want to know the sequence. So this is there. Here you can see the DNA sample which is having a 5' and 3' end. The important thing is that at 3' end there is always a hydroxyl group, and at 5' end there is always a phosphate group. So this DNA sample, you have just one molecule I have shown here out of the DNA sample. From this DNA sample we'll find out the base sequence. In this case, what we are going to do, the first thing, that we are treating this whole thing with the alkaline phosphatase enzyme. And why we do that? To remove the phosphate. And why we have to remove the phosphate? It is because we have to do the radio labeling of this molecule. It is very important to do the radio labeling. Any kind of labeling should be there, and in this method we do the radio labeling. And for doing the radio labeling, what we do, we have to use some radioisotope. So here we use the radioisotope of phosphorus. In the presence of kinase, we have given P32, which is a radioactive phosphorus, not the normal one, and the radio labeling will be done. So how the radio labeling and where it will be done? Phosphorus is present at the 5' end, and here you see you have already removed the phosphate from the 5' end. So from the 5' end, the normal phosphorus was removed. Instead of that normal phosphorus, here the radioisotope will be added. So this 5' end and this 5' end is now having the phosphorus which is radioactively labeled, and this radioactivity I have shown by the asterisk. Now you see we have to do the denaturation. Denaturation, you already know, that means we have to make the DNA as the single stranded. This was double stranded. From the double stranded we have made this DNA single stranded, which is having a 5' end, which is radioactively labeled. So this is something which is very important, and denaturation can be done at the high temperature. Next is that we have to divide the sample into 4 parts, because ultimately we are going to do this whole reaction in 4 different tubes. In all those 4 different tubes we'll put some chemicals, some different chemicals. We'll add those chemicals and they will do some reaction. So that's why this DNA sample, there won't be just one DNA molecule, there will be lots of samples. So that will be divided into 4 parts. Now let's see what we do next. Here I have shown the theory of the chemical degradation. You know that here chemicals are very important in Maxam and Gilbert method. So what actually they do, what is the principle? So that we want to discuss now. So this is a two-step catalytic process. In the first one, you see the chemical or the chemicals, means like a combination of two chemicals, and they break the glycosidic bond between the sugar and the base. You see, this is the base at this carbon position first, and this is a ribose sugar. It's a diagram of a nucleotide, and here is the position of glycosidic bond. So at this point, these chemicals act upon and they displace the base, because base is attached to this, so this base is dislocated. So that is what is done by the in the first step. And here also one thing important here, that there are certain chemicals which we are going to use, they act only upon the purines, and the other group of chemicals will use which only act upon the pyrimidines, CT, and purines are AG. The chemicals which we use are dimethyl sulfate, hydrazine, formic acid, and NaCl. So this is the first step out of the two steps. Then let's see what is the second step. In second step, we are going to use piperidine, and this piperidine we are going to use whether it is purine or whether it is pyrimidine. In all the cases, we'll use piperidine, and it will catalyze the phosphodiester bond and make the final break. See, in this is a polynucleotide chain. I have shown only two nucleotides, but it must be the long chain. The first nucleotide is here. You see, the base has already been replaced because of this first step. But phosphate is still there, and at the OH hydroxyl group, this is the 3' end of this 3 carbon sugar, and phosphorus of this particular nucleotide will get attached to it, and a phosphodiester bond is formed. Although the base was displaced, but it is still attached to the polynucleotide chain. So what is done in the second step? At the second step, this phosphodiester bond is broken, and that's how this will get detached from this one. So that means here the chain will be broken. So degradation will take place, and because this degradation is taking place because of the chemical, so that's why it is chemical degradation. So these are the two steps. Means ultimately at a particular base, we know which base has been displaced, and we know that the link has been broken from this point. The polynucleotide chain has been broken from this point. Now we'll see what are the chemicals and what is their role. I told you that we'll whatever single stranded sample we have, will we'll divide it into 4 parts. So in the first tube we see what is happening. In the first tube we call it G reaction, and I'll tell you why it is known as G reaction. In the first tube we will add DMS. DMS is dimethyl sulfate, commonly known as DMS, and piperidine. Piperidine, I told you that in all the 4 tubes we'll put the piperidine. So this dimethyl sulfate, or which is commonly known as DMS, it acts upon purines only. So it will act only on A or on G. Now if you use only DMS, then what it will do? It will modify G, and ultimately you will use piperidine. So at this point, because the modification has been done in G, so at that point the removal of nucleotide will take place and the chain will be broken. So this is the first tube, and that's why, because we know that the chain has been broken at G, so that's why it is known as G reaction. Now you see what happens in the second tube. In second tube we have added DMS. Along with that we have also added formic acid, and then there is piperidine. Now this we are calling as A plus G reaction. So here what is happening? DMS we have added, we already know it acts upon the purine. Then we have added formic acid. Formic acid breaks the link between purines and sugar. That means both the purines, A and G. So between these A and G bases, purine bases and sugar, the glycosidic bond is broken because of the formic acid. So that's why we are calling it here A plus G reaction. And then of course the piperidine, you already know that it makes the final break. So this was the reaction in the second tube. Now let's come to the third tube. In the third tube we have used hydrazine and piperidine. Now hydrazine acts on the pyrimidines. So there are two pyrimidines, C and T. So if only hydrazine is added, then what it will do? It will open up the ring and hydrolyze not only C but also T. So both C and T both will be hydrolyzed. And then piperidine, as you know, that it will cleave and it will break the chain. Now let's see fourth tube. In case of fourth tube, hydrazine was added. Along with the hydrazine, 1.5 molar solution of NaCl was added, and then there is piperidine. What it does? Hydrazine, of course, it acts on the pyrimidine. Then because we have added NaCl also, so what will be done? NaCl inhibits the reaction of T. So only the C reaction will take place. That's why you can see C reaction here. And then role of piperidine, you already know it will finally cleave the chain. So this is the, these are the four reactions which are taking place in presence of four different chemicals. Now we'll talk about what is the procedure after the chemical degradation, how we are going to find out the sequence. We already have a single stranded DNA which we have labeled also at the 5' end, but mostly the single stranded DNA which we have, not mostly, actually always, we do not know the sequence. We have to find out the sequence, but here, just to make you understand, I have written a sequence here. Now let's see what happens in the first tube. In the first tube was the G reaction, and in which we added DMS plus piperidine. So all the reactions will take place here. Now you see, because it is G reaction, so where all these chemicals are going to attack? At the G. Which will be dissociated? The G will be dissociated or displaced. So now you have to see, in this single stranded DNA, G is present at this point and at this point. That means there are two G's. So what will happen? When these two chemicals will be added, they will break the chain at this point, just before G. And the second point is here. They can also break the chain at this particular point. So if the chain is broken at this particular point, what you will get? ATTC. So here you can see ATTC, and at the 5' end, at this end, at the 5' end, it is radio labeled. Or in the second case, you get ATTCGC. Up to this point, you will get the fragment, and here also you will see that at 5' end there is radioactive labeling. When the chain will be broken, there will be one more fragment. One fragment would be this one, which has this 5' end. There will be one more strand or one more fragment, but that fragment we are not going to use, because, and how we will know? We'll know by the radioactive labeling, and that's why doing the radioactive labeling was very important in this case. So now let's see what happens in the second tube. In the second tube, the reaction was AG, and this was the combination of the chemicals. Now AG, what does it show? It shows that these chemicals will not only cleave A, but they will also cleave G sometimes. So they will cleave A. Wherever there is A, they will cleave A, but also G to some extent. Now again see, here the A is the first one. But if the reaction is cleaved at this point, then what will happen? You will get just this 5' and phosphorus. So, this nothing, it doesn't make any sense. Then see, where is A? A is present here also. So in this case, it will act upon this particular point, and you will get a fragment from this point to this point. So here you can see, this is the one. And then A is present here also. So then the break will take place at this particular point, just before A. So in that case, you will get a fragment right from this to this. So that is present here. So this was about A, but then you have to see the G also. Now where is G? G is present here. So you will get the breakage at this particular point, ATTC. So this you will get, and the another G is present here. So again it will break at this point. So you get ATTCGC. Similarly, third tube also, the reaction chemicals are different, and it is C plus T reaction. So this will break the DNA at C, wherever there is C. So at these 2 points the breakage will be there, and wherever there is T. So T is at present at this point, at this point, at this point, and at this point. So at all these points you will see the break will take place. So first, you see, if the break was at this particular point, you get ATT, and then something like you can see how you get these different fragments here. And then in the last tube is known as C reaction, and the combination of the chemicals which you have used is this particular hydrazine NaCl piperidine. And here in this case, you see it will break the chain only at the C point. So there are 2 times C here. So one break is going to be at this point, and other break is going to be at this point. So once along with the 5' and you get this ATT, and second time you get along with the 5' and this up to G. So these 2 fragments you are going to get here. So here in this case you will get 2 different type of fragments. In this case you will get 6 different types, 4 different types, and 2 different types fragments you will get here. And then we'll see that how we'll do the interpretation, we'll see. And then ultimately you will be able to get the same sequence, only except for the first base. Right from the second base we'll get the sequence. Let's see what happens next. After this chemical reaction has taken place in 4 different tubes, you have to do the electrophoresis. Here you can see we have shown only 4 lanes here, 4 wells I have shown you here. In the first lane, well, you will put the product of the first tube. In the second lane you will put the product of the second tube, then third and fourth. The principle of electrophoresis, you all must be knowing that these DNA, when they separate, they start traveling on the gel, and when they travel, the lower molecular weight or the bands which are very small in length, they will travel fast. So they will move far from the well. But if they have the higher length and the higher molecular weight, then they will remain somewhere near this side. So lower molecular weight ones would be somewhere here, and the higher molecular weight would be somewhere here. And then after the electrophoresis, you have to do the autoradiography. Autoradiography here is very important because that's how you are going to find out your fragments. Why? Because you have done the radio labeling here. So because your DNA is radiolabeled, so you have to see the image on the X-ray film and you will get the autoradiogram. Once you get the autoradiogram, then you have to do the interpretation of bands. Just see carefully, it's not that difficult to find out the bands. Here in this lane you see the G reaction, two products are there. Here A plus G is there, C plus T and C. Now you see, where is the first band? At the bottom. The first band is somewhere here in C plus T. So you see, this band is present only here, not here. So that means it is showing T. Then the second band is also present in C plus T. So that means it is also going to be T. But here you see, this band is present at the same position in this lane as well as in this lane. That means this band is present in the product of this tube as well as in the product of this tube. So that means what it is showing? It is showing that it cannot be T. It should be C, because C is present here as well as here. C is common in both. So that's why this band is also common. So interpretation is that it should be C. It can't be T. Had it been T, it should have been present only in this particular lane. Now see what is next. Next band is present here. But it is present in both the lanes. Now you see in both the lanes what is common. So here you can see the G is common. So that means it should be G. Then again C, because it is present in both the lanes. So C is common. So it should be C. This should be again G. But this is present only here. So this should be T. And then this should be A. Then again T. And then again A. So that's how you can find out the sequence. Right from the 5' and to 3' and it can be read from this side to this side, T, T, C, G, C, G, T, A, T, A. So that's how we interpret the bands. There is one more way of writing this sequence and interpreting this, and this is, I prefer this one. Here you see the same band pattern which was in the previous slide. Same. This is T. Here I have shown the 5' and which is already radio labeled, that, so there is only this T. Then again this is also T. So here I have shown 2 times T. Why? Because T was already there. This was the smallest band which was having only one nucleotide. But this is a little bigger than this one. This is little bigger. So this is made up of 2 nucleotides. So one nucleotide you have already interpreted. You already know it was T. And the second is also T. And this third one we interpreted, it is C. So T and T were already there. And here you can see this last one is C. Then this was a little higher molecular weight, having 4 nucleotides. So here you see TTCG. So that's how the size will be increased, and you see from 5' to 3' and this is the sequence of the single stranded DNA. This one. And this sequence is same. If you remember, we supposed that in single stranded DNA there is this particular random sequence. So you check it with that. It is same except for the first nucleotide. As I said, that cannot be interpreted from Maxam and Gilbert method. So friends, that is all about the Maxam and Gilbert method. And I already told you that now the next generation sequencing methods are available which are much more advanced. So these days if we have to do the sequencing, then we go for those methods. So thank you so much. And if you like this video, then please subscribe my channel so that you can see my other videos as well.